Hey guys, it's Renee. Every 10 minutes, another person is added to the waiting list for organ transplant, and 22 people die each day waiting. Even though the demand for organs is high, the organs available are simply not enough. However, what if I told you that there is a solution to solve all these problems? My concern in the low supply of organs have inspired me to do this topic. Today, I'll be talking about human pig embryos, the first step in growing human organs in pigs. Before we start talking about the actual process, let's first explain some keywords. So what exactly is a human pig embryo? Well, just to clear confusions, a human pig embryo is simply a pig embryo, which is a fertilized egg forming important body systems containing human cells. The second keyword is chimera. Chimera is an animal composed of two different genomes. By this it means an animal contains two different sets of DNA in different cells in its body. The word chimera originates from Greek mythology. It's a creature that's composed of body parts of different organisms. This creature is pretty much an extreme image of the word chimera. Because there are different sets of DNA in different cells, body parts of different organisms grow. The lion head contains lion DNA, the goat head contains goat DNA, etc. Remember that all DNA in your cells are the same. It's just the sections that each type of cells read are different. But for a chimera, which in this case is a pig, it contains pig DNA in a pig cell and human DNA in a human cell, and both these cells are present in the pig. Another mythological example of a chimera are angels, which is a chimera of humans and birds. The body contains bird DNA along with human DNA, which causes wings to grow. Now with these knowledge in mind, let's move on to the actual process. The process of creating human pig embryos starts with generating pluripotent stem cells from a human somatic cell. A stem cell is an unspecialized cell, which the cell is not programmed to signal proteins to reach specific DNA sections, so they can turn into any type of cell. There are three types of stem cells. So now many of you may question, how is it possible for pluripotent stem cells to get generated in the human body? The only type of stem cell that can be found naturally in certain parts of the human body, such as the bone marrow and the heart, is multipotent, which means it's only able to turn into specialized cells of the same category, such as B cells and T cells, which are both white blood cells. However, scientists have found a way to generate pluripotent stem cells that have the potential to turn into any specialized cell from any somatic cell of our body. This requires reprogramming to create IPS, induced pluripotent cells. The word induced simply means to cause something by stimulation or influence, which in this case, scientists act as an outside force to create pluripotent stem cells through reprogramming. Scientists question whether if proteins that make embryonic cells pluripotent are able to reprogram a specialized cell, turning it into a stem cell again. They began with a list of 100 possibilities, which created over 1 million variations. With a computer program, they found the 4 essential factors. They took these 4 factors that acted together in embryonic stem cells and inserted them into a somatic cell. In a process we don't fully understand, parts that were normally tangled and winded since certain sections are not read in certain somatic cells began to unwind. Factors can now attach to a gene, transcribing it and produce embryonic cell proteins. Large amount of these proteins are made, giving signals and messages to fool the cell to believe that it's in a place that requires it to become an embryonic cell. The reprogrammed cell replicate and become more like pluripotent stem cells until they finally become one. The stem cells are later inserted into a pig embryo using a tube, as shown as this picture. The surviving embryos are put into adult pig's uterus for development and hopefully create an organism that shows chimera, which contains human IPCs and pig cells. The embryos are later removed from the pig's uterus and analyzed. More than 186 out of 2,075 embryos continue to develop. The embryos only contain a small amount of human cells. Around 1 in 10,000 of the pig cells were human. It is obvious that pig cells greatly outnumber human cells. The following bar graph is a graph on chimera between rats and mouse of a separate experiment. The green part is the percentage of the organism aborting the recombinant embryo before birth. The red part is the percentage of non-chimeras, which the embryo did not contain cells of another organism. The blue part at the very bottom is the percentage of actual successful chimeras. It's obvious that fails overwhelmed success. Even though this graph is not on the actual human pig embryo experiment, this can act as evidence for us to infer that success rates for human pig embryos might be low as well since they're both chimeras. This low rate of success may create problems. Since the cells only contain a little amount of human cells, it's still unknown whether if a pig can tolerate a larger amount of human cells in their body. There are also ethical concerns. Whether if the recombinant pigs will have human cells in their brains, making them get human qualities, or will they have human cells in their productive cells, producing human eggs or sperms, or if they'll pass down human traits, are all questions that still remain unknown. Also, if the transplants are possible, it might blur the line between humans and beasts. If pigs, who we humans have seen as animals for food, now become heroes for growing organs and save human lives, will our relationship with these animals change? One interesting thing to think about is, if you've received a heart transplant from a pig, or if your friend or family member currently has a functioning pig's heart in their body, are you still able to eat bacon without scruple? Despite the cons, there are still pros. Creating embryos with human cells allows scientists to screen drugs before human trial, decreasing the risk of drugs endangering human health. It also allows scientists to study how genetic diseases develop, and perhaps learn ways to prevent it. It also allows us to understand early stage of embryo development, and of course, harvest organs for transplants. From the graph shown here, the number of people waiting for organ transplant has increased dramatically, whereas the number of available transplants have remained the same, not meeting the needs. Creating human pig embryos will greatly reduce the deaths of people waiting for transplants and lower the rejection risk of transplants. The patient's immune system won't attack the new organ since the organ is generated directly from the patient's stem cells. Even though the actual human organs have not been harvested yet, the experiment of creating human pig embryos is a huge breakthrough. The insights that we learn while researching and experimenting are just as valuable as the organ itself. Hope this video made you understand more about recombinant organisms. Thank you for watching.